Welcome, guys. I salute all of you. Today I want to start a series of videos on biology practicals to help those who are preparing for the KCSE examination. I would like to inform the viewers that I was part and parcel of coming up with the polished documents as far as the marking schemes were concerned, because I've been an examiner from 1989 to roughly 2003, when I was promoted to be a senior examiner, and therefore whatever I'm going to produce has been polished, and you can bet with me that if you watch this video keenly and follow the slides, you are there to score a very good mark in biology paper 3. So, welcome guys to my paper 3 series, KCSE examination. So guys, I'm starting with the forward of the videos. I'm trying to adjust the one. What has happened? So guys, welcome. I started marking in 1989, but I'll start my presentations with a paper from 2001 to see the response. So guys, I welcome you to this channel. Comment on this inaugural video on biology practical. So I start with the foreword, where I say this video has been made to assist biology secondary students in paper 3 biology practicals. It covers the entire biology KCSE syllabus, and its preparation has been done by Midhamoti K, a biologist, a biology teacher, and a biology examiner of long standing in Kenya. I'm not trying to blow my trumpet. This is true, guys. He has been a senior examiner and senior HOD sciences at Kerugoya Girls High School for more than 10 years. He has earned the honor of being the best biology teacher in Kirinyaga County in 1997. So this video is being handled or being produced by somebody who knows what is what. So let's move. So, disclaimer. This is an educational video. Copyright disclaimer under Section 107 of the Copyright Act of 1976. Allowance is made for fair use for purposes such as criticism, comment, news, reporting, teaching, scholarship, education, and research. The material used in this video was available to me by virtue of being an examiner and a senior examiner from 1989 to 2016. And in fact, I was also a trainer of examiners from 2015 to 2017. I was training the examiners for biology practical. So I've decided to start with the papers of from 2001 to 2016 because that is where I retired. But if anybody requires the other papers, I can also access them. But mostly what I've seen is that most of these papers have got the same concepts being repeated once and again. So concepts are not different. So if you are keen with what I will be giving you, you'll be lucky when it comes to the real examination. So in 2001, it was called paper 2, but now they have changed it and called it paper 3. Question 1, you are provided with a portion of onion bulb. Remove one fresh leaf from the portion. Peel the epidermis from the inner surface of the leaf. Place it on a drop of water on a slide. Place a cover slip on the epidermis. Place a drop of iodine solution at one edge of the cover slip. Using a blotting paper, drain off excess iodine solution and water from the opposite edge of the cover slip. And then observe the epidermis under low power, then under medium power of a microscope. So in this question, you had been provided with a microscope with low power and medium power. So You are told, part A, draw and label 2 neighboring cells, and you are being given 5 marks. So you can see now how the marking was done, and you can also see the drawing of the 2 cells, at least 2 neighboring cells. All right. So after discussing the marking scheme, the senior examiners came with that final product. You can also see what was being rejected, which is in red. If the reject diagram, if only one cell is drawn, so everything could not be marked. Reject also labels with arrows or conspicuous dots at the end of the label line. So the label line should have been complete. And again, reject wrong spellings for labels. Then there is a place there where you are being told to allow pentagonal, hexagonal, rectangular shape. I think you see these things as we go to the other questions. And then mark if more than 2 cells, okay? Label lines should touch the structure. Adjacent cells should not be more than 1 millimeter apart, okay? Then accuracy mark. Outline is continuous. Nucleus placed at side, not center. No shading except nucleus only. Cells must appear elongated. Double line. More than 1 cell. No use of ruler. So if you look at the diagram there, you can see the labels there. We have marked the cell wall, right? The cell membrane, the cytoplasm, and the nucleus. The magnification mark could be any of those magnifications given there. Let me try to adjust this a bit. All right. Times 20, times 25, times 40, times 50, times 25, times 100, times 150, times 225, times 225, and times 90, except if somebody says 90 times or 50 times or 20 times without necessarily putting the X. The magnification must be preceded by a capital X and not a small x. Question B was, why was staining of the epidermis necessary? And the answer, which the examiners agreed on as a final marking scheme, was to make different parts or various parts or various organelles of the cell distinct. So here you may say to make different parts distinct, to make various parts distinct, to make various organelles of the cell distinct, or to make different parts clear, or make different parts visible, or make different parts conspicuous. Any of those ones was around. Work out the length and width of one cell as seen under medium power, where you are being given 6 marks. So follow here. You are supposed to get the diameter of field of view which was between 1 to 2 millimeters. Your teachers have taught you in Form 1 how to get the diameter of field of view. So the range was between 1 to 2 millimeters. And therefore, you are going to multiply this range. If it is 1 millimeter, you multiply with 1 micrometer, the units of microscope, microscopic units. And if you multiply with 1 millimeter, if it was 1 millimeter, with 100 micrometer, you get 1,000 micrometers, and so on, right? C, work out the length and the width of one cell as seen under medium power. So the number of cells in the field of view were ranging from 10 to 20 across the width, and they were ranging 3 to 5 along the length. So width of one cell was 100, I'm giving the range, 100 to 200 over 10 to 20, depending now on what you use, and you are getting the answer as the width to be between 100 to 200 or 50 to 100 micrometers. Work out the length and width of one cell as seen under medium power. The length of one cell would be, you divide now the micrometers you have got by 3, if you got 3 cells, or 5, if you got 5 cells, and the answer was 3333.3 micrometers to 666.6 micrometers, depending on what you get, or 200 to 400 micrometers. So you get 8 marks. The marks are the commas, the semicolons are the, in marking, a tick is represented by a semicolon. So if you count the semicolons, you find that there are 8, but the maximum mark was 6. Question 1D. Place and drop of liquid air at the edge of the cover slip. Drain the liquid from the opposite edge to allow it to flow across the epidermis. Leave the setup for about 5 minutes. Observe under medium power. After observing, you are told, draw and label 2 neighboring cells. Okay, so it's now after adding whatever you are being told to add. So mark D and E independently. Benefit of doubt. Treat the diagram as in A. All right, the diagram should be treated the way we treated A. All right. Accuracy mark. No shading except nucleus. Nucleus must be, let me adjust, please. Let me adjust, yes. Accuracy, no shading except nucleus. Nucleus must be in cytoplasm, outline must be continuous, vacuole if present must be very small, cytoplasm must show shrinkage. Then you have the labeling here, cell wall, cell membrane or plasma membrane, cytoplasm, and nucleus. Account for the results in D above. So this is what you are accounting. You are getting 4 marks. Liquid L is hypertonic. Water is drawn out from the cells by osmosis, making the cytoplasm shrink. So, when you say liquid L is hypertonic, you get a tick. Water is drawn out from the cells, a tick. that is the semicolon, by osmosis are thick and making the cytoplasm shrink, a thick, or making the cytoplasm cell membrane shrink, the same, or making the cytoplasm plasmolyzed. So you find there, you have how many marks? 4. And then we have OWTT means or want to that effect. The alternative to the answer we have given above was liquid L is hypertonic. Instead of saying hypertonic, you could say more concentrated than cell sap, or once to that effect. You get a mark. Water is drawn out of the cells. You get a mark. By osmosis, or some people will say by osmosis, but me, I prefer you say by osmosis, you get a mark. Making the cytoplasm, all cell membrane shrink, all plasmolyzed, okay? All withdrawn from the cell wall, making the cytoplasm or cell membrane shrink, plasmolyzed, withdrawn from the cell wall, mark. So here, what you do is mark the points independently, okay? If you fail by osmosis, don't worry. You mark them independently. Reject flaccidity because you don't feel. Liquid air absorbs water. Shells shrinks. Vacuum shrinks. So don't entertain anyone like flaccidity. The cells become flaccid. There was no, you are not touching. This was microscopic work. Question 2 of the KCSE practical paper was, you are provided with specimens labeled M and N. Identify the specimen and state the organism from which they were obtained. So let's see. One was 4 marks, and in drawing the specimen, M, you are getting 5 marks. So let's see. So, specimen M was gill, or fish gill, or gill from a fish. So the organism is fish. Reject if they give any specific name of a fish, like saying gill, organism, you say tilapia. No, don't mention specific, just say fish. N is a piece of lung, but you can say it's lung or lung tissue. Organism, mammal, or mammal, e.g. goat, cow, sheep, right? Remember, A is tied to E, the E which is coming. If you fail A, you cannot get E, all right? So you don't even mark E. So A is tied to E and D, but if identity is correct and organism long, proceed to mark E. See, there are technicalities in marking. That's why you find people in paper 3 have issues, and they need to be trained by an examiner to know the tricks. Accept mammalian instead of mammal. Reject mammalia because it is a class. Reject pieces for fish because that is a class. Avoid, I want no mark for organism if identity is wrong. So if somebody does not get the identity, you cannot mark for the organism. All right. So draw and label M. All right. So it is there. Mark A1 and A2 independently. Labels should be given. That is accuracy. Labels should be given correct name, should be correctly spelled. All right. I will adjust this for the other points. Gill filament, gill bar or gill arch, gill raker. You get labels are 3, accuracy 2 marks. And you can see where the accuracy is being given. You see the way the arc is bedding, all right, and the gill bar. So after adjusting, we have now what we have there. The labels are very clear, but there was this down here, how you are giving the first accuracy. Accuracy 1 is for continuous outline. All parts of the gill drawn properly, no shading. Okay? Accuracy number 2, all the 3 parts drawn proportionally in correct sizes in relation to one another. Curvature of the gill bar must be shown. Gill rakers should not be close to each other. For you to get the second accuracy mark. So the first one is continuous outline. All parts of the gill drawn properly and no shading. You can get to that one. You can see where it is being pressed. The second one, A2, all the 3 parts drawn must be proportional. You don't have a gill raker which is bigger than the gill bar, or the gill bar bigger than the gill filament. All right, guys. So, using observable features only, explain how specimen M is adaptant to its function. And there we have given, there you are getting 8 marks. And you have numerals. Instead of numerals, you can say many. And instead of many, you can say several. So, to read like this. Numerous gill filaments to increase surface area for gaseous exchange, or numerous gill filaments to increase surface area for absorption of oxygen, or numerous gill filaments to increase surface area for release of carbon dioxide. Then we have rigid gill bar or rigid gill arc to provide firm attachment, or to provide attachment. Where you see the bracket, it's not necessary to say those ones. You can say gill bar to provide attachment, to hold, okay, to support gill filaments or gill rakers. Next point, presence of gill rakers to prevent solids from reaching the delicate gill filaments. So you can avoid the word projecting. You can just say gill rakers. Thin filaments to reduce distance from which gases diffuse. Long filaments to increase surface area for gaseous exchange. So each point there, each bullet has got 2 points. And if you count them, they are 1, 2, 3, 4, 5. So there are 10 points or 10 marks, maximum 8. D. State 3 distinguishing features of specimen N, 3 marks. Presence of bronchus, or you say alveolar ducts. Pleural membrane, spongy, air sacs, sponginess. All these will give you 3 marks. So presence of bronchus, presence of pleural membrane, the specimen being spongy, or the specimen having air spaces. Reject if somebody talks of air sacs, or if somebody talks of sponge instead of spongy. E. State the functional relationship between specimen M and N. Gaseous exchange, or you just say it is a respiratory organ, and you get your 1 mark. Reject breathing, respiration, or gaseous exchange. You say breathing, then in brackets you say gaseous exchange. Those ones are going to be rejected. It is for gaseous exchange, respiratory organ. If somebody puts the word breathing there, 0. Question 3. You are provided with the specimens labeled P1, P2, P3, P4, P5, and P6. And a dichotomous key shown below can be used to identify them. So you are given the dichotomous key. If you find leaves are simple, you go to 2. I hope by now you have been taught how to use the dichotomous key. Leaves compound, directly you identify that one as acacia. Leaves green, you go to 3. 3 is coming. Leaves purple, your identity is Tradescantia. Leaves parallel veined, Z. Leaves net veined, go to 4. Leaf margin serrated, go to 7. Leaf margin smooth, go to 5. Remember, dichotomous means comparing 2 comparisons. Die 2. Dichotomy, the comparison. 5a, leaves hairy. That one is Solanum. Leaves not hairy, you go to 6. Leaves ovate, Bougainvillea. Leaves lanceolate, Mangifera. Finally, leaves fleshy, go to 8. Leaves not fleshy, hibiscus. Leaves with pointed tip, kalanchoe. And leaves with rounded tip, Bryophyllum. So you have identified all the organism. So the question is this. Use the dichotomous key to identify each of the plant species provided. In each case, show in sequence the steps 1a, 2b, 5b, etc. in the key that you followed to arrive at the identity of each specimen. So using the dichotomous key to identify each of the plant specimen provided, steps specimen P1, P2, P3, P4, P5, P6, steps followed. For you to get Bryophyllum, Bryophyllum, you are need to go to step 1, 1A, 2A, 3B, 4A, 7A, and 8B. You identify Bryophyllum. P2, 1A, 2B, Tradescantia. P3, 1A, 2A, 3B, 4B, 5B, 6A, Bougainvillea. P4, 1A, 2A, 3B, 4B, 5B, 6B, Mangifera. P5, 1A, 2A, 3B, 4B, 5A, Solanum. And P6 is steps are 1A, 2A, 3A, which is Z. And in that case, you have identified all the specimens given to you using the provided dichotomous key. Maybe I'll mention what was there in the confidential after I finish this. So in equation B1, after the identity, name the likely habitat of specimen P1. P1, of course, you have already identified it, all right? And its habitat is arid. You say arid, or you say semi-arid, or you say desert, or you say dry area, or you say dry land, or want to that effect. Reject if somebody says hot land or hot area. That one will be cancelled. B2, give a reason for your answer in B1 above. The reason is that it is freshy, or it is succulent leaves. Fresh leaves, or succulent leaves, or just fresh or succulent. Juicy, it is juicy, or it has thick cuticle. Any of those answers. When you see the stroke means the answers are equivalent. And therefore, you reject fresh, or fresh, or fresh, or fresh. Those ones will be rejected. You know, people have problems with mother tongue and they write wrong biological terms. C, state the significance of the shiny upper surface of specimen P4. To reflect away sun's rays, to reduce water loss, or to reduce transpiration. I'm saying strokes means you use only one hand. When answering in KCSE, students have problems after being exposed to marking schemes. They give answers with strokes. Please avoid. Just choose one word. To reflect away sun's rays, to reduce transpiration, or to reflect away sun's light, to reduce water loss or transpiration. All right, be careful. And then to reduce heating up of the reef, to reduce transpiration, or to reduce water loss. So, D, observe the floral parts of specimen P3. What is the significance of the brightly colored structures onto which the flowers are attached? You get 1 mark, and you have the answer as to attract insects for pollination. Okay, you don't need it to say for pollination, but it is good. You are good biology students if you say to attract insects for pollination. Reject to attract pollinators. That one will be rejected. E. Question 3E. Name two features that makes specimen P5 adapted to its environment. 1. It is hairy to reduce water loss. It has hairs or it has hair. Any. You can say anything there and you get a mark. Presence of thorns, spines or spikes or prickles. Any of those ones, you select one to prevent herbivorous animals from feeding on them. So any of those 2 points will give you 2 marks. So if you say hairy to reduce water loss, you get your 2 marks. Or you say the presence of thorns to prevent herbivorous animals from feeding on them. F. Name a feature that is used to classify P6 as monocotyledonous plant. 1 mark. And there, the straight answer there is venation, or you say parallel veins, or you say veins, all right? Okay? Or you say arrangement of veins, or you say presence of sheath, or you say leaf sheath. Any of those ones will give you a mark. So I will finish this video by acknowledgments. It is my pleasure to say this. This work is truly a team effort. There are too many people who inspired me to come up with this production of this video, or with the production of this video. And I thank them with all my heart. I would also thank my family members for their support, encouragement, patience, tolerance during my long hours of typing the PowerPoint presentation. I'm also grateful to all my teammates during marking exercise of the KCSE Paper 3, who in one way or the other participated in making coming up with the final draft of the marking scheme during our modulation as senior examiners. Thank you very much, guys. Subscribe to this channel. Give the video a like so that when you hit that button, notification button, I'll be able to give you more videos of this type. All right? So guys, I have nothing else to do but only to thank you, and I ask you to watch this video from the beginning to the end if you have a chance of coming across it wherever you are in this world. Thank you guys for watching my videos, for subscribing to my channel. Continue also encouraging your friends to subscribe to my channel so that you support me to come up with such videos. Guys, goodbye and see you next time in my next video. And the year will be 2002 practical.